Good morning. I'm Gary Ritchie for Pharmaceutical Manufacturing Magazine. I'm here today at IFPAC 2009 with Dr. Stefan Mallet from Pharma LIBS. Stefan is Pharmaceutical Vice President of Product Development, Business Development for Pharma LIBS,
yes.
And he's here today to talk about the Pharma LIBS 250.
Absolutely. Thank you very much, Gary. Pharma LIBS 250 is an instrument that we manufacture. Pharma LIBS is a spin-off company from the National Research Council of Canada. We basically are focusing on the LIBS technology, which is laser-induced breakdown spectroscopy. Our instrument is an instrument designed specifically for pharmaceutical applications. It will analyze in seconds the content uniformity, dosage determination, coating thickness uniformity of a tablet, or you can also analyze the content uniformity of a powder. But all for solid dosage form. The way it's designed, the system is for solid dosage forms. Mm-hmm. The biggest advantage of that technology is that you don't need to sample prep at all. So it's very simple. Put the tablet in the system, plug and play.
Analysis. First of all, the fundamental basic understanding of LIBS. I am new to that area, and I'm sure certain people, members of our audience, are too. Perhaps we can get a little background on this from the Chief Science Officer?
Absolutely. We're fortunate enough to have today Dr. François Doucet with us. François is with the National Research Council of Canada, and he is part of a team that are developing the LIBS technology. Quite an expert, actually, and we're happy to have him on board. I mean, have him as a scientist, as a partner in that new adventure with LIBS.
We'd love to hear from him.
Dr. Doucet.
Good morning, sir.
Good morning, sir.
It's good to see you. LIBS. Give me a little understanding, and our audience a little understanding of, for instance, the analysis for pharmaceutical purposes. We do content uniformity, we do assay, we do dissolution, we do identification. Could LIBS be used to do any of those analyses?
Yeah, for sure. LIBS is a spectroscopic technique based on the physics of atomic emission spectroscopy. So the only difference is the generation of the source of energy to generate the plasma, which is a laser, pulse laser in that case. So typically you focalize a laser pulse on the sample to generate a plasma. So you ablate material, you generate a plasma, and this plasma is information rich of what you sample on your
We have a molecule, so we're getting molecular information?
It is possible also to get molecular information about your sample, especially when you're using the laser-induced molecular bands that are produced in the generation of the laser, along with chemometrics. So you are able to extract all the excipient inside as well as the API. Additionally, you can pick up the hardness of the tablet and if it's a wet granulation or direct compression.
How would this technique lend itself to process analytics?
I think that one of the Graal application of LIBS is the max T rate. For content uniformity. It's really efficient, need no sample preparation, means that there is no chance that you contaminate your sample. Mm-hmm. So you are able to generate a 3D map. You can even make disappear your tablet if you want to generate a more complete mapping of the max T rate inside your tablet. What else? You can do it for other components also, using the molecular LIBS part, where you use chemometrics. You can then apply the same sampling technique in order to generate a map of your API or your excipient, if you want.
That's very interesting. It's very good to speak with you today. I think I now understand a little bit more of LIBS, and I think I'd like to wind up the discussion with Stefan. Thank you very much.
Thanks, François. Thank you.
So, Stefan.
Yes.
Where have you? What's your future look like with the application? Do you have any applications you could talk about now?
Yes.
White papers, technical notes, any of those things?
Absolutely. I think what Francois covered is one of the most important applications of this technology, magnesium stearate distribution inside a tablet. Because basically the way it works is once the laser ablates the material, it's going to make the molecule explode, basically. Mm-hmm. So magnesium stearate has magnesium at the surface, obviously, and this is what we're tagging. This is what we're looking for in the molecule. Mm-hmm. So imagine any type of API having a tag on it. It could be chlorine, it could be magnesium, it could be potassium, it could be sodium, and you can also look at the aromatic cycles that you have inside an API. Because once the laser hits the aromatic cycle, there's still C2 bonds remaining. So we can look at the emission band of C2s. Mm-hmm. So basically, with this atomic emission spectroscopy, you can look at approximately 80% of all the APIs that you have out there.
Many of the pharmaceutical houses are interested in solving some of the complex problem related to the performance characteristics of their products. Give us an example of LIBS technology solving issues in processing with respect to, let's say, a low dosage formulation for uniformity issues or dissolution? Okay.
Well, basically, what we can solve, the problem that we've been solving with pharmaceutical companies as troubleshooting was looking at exactly what you're saying, low dosage concentration of an API. Mm-hmm. Because this technology has the advantage of going down to low ppm, high ppb levels for some of those APIs. So if you compare it to other current technology or conventional technologies, well, you can go a lot lower. And then you can basically do this without any sample preparation. This is something that I need to remind you.
No sample prep.
It's very easy. You get the results in seconds, and you go all the way down to the low ppm levels. So that's one thing. The other thing that is very interesting in terms of an application, we've been doing lots of research with Rutgers University, and Dr. Fernando Muzio did a presentation here at IFPAC yesterday about coating thickness uniformity. So what the instrument is capable of doing also is to look at the uniformity of the coating by tagging either titanium present in the coating, which is usually present in the coating, or the number of pulses needed to go through the coating until you hit magnesium, which is always or almost always present inside the core of the tablet for magnesium stearate. So we were able to demonstrate very clearly that coating thickness uniformity can be very well identified with this technology.
Well, this all sounds very exciting. It is a pleasure to speaking with you today.
Thank you very much.
IFPAC 2009. We look forward to seeing you back in the future with perhaps newer development in this field.
Absolutely. Thank you very much, Gary.
Thank you very much.